Hello, this is Dr. Patrick Hu, President of SITC and President and CEO of Moffitt Cancer Center. And I'm really happy today to have as our guests Dr. Mario Sznol and Dr. Jedd Wolchok. Both have been in the immunotherapy field for many decades and have had lots of major contributions, including the development of one of the most important combinations in immunotherapy today, Ipilimumab and Evolumab. Dr. Sznol is head of translational research for melanoma and kidney cancer at Yale Cancer Center, and Dr. Wolchok is head of immunotherapy and melanoma at Sloan Kettering Cancer Center. So welcome, you guys.
Thank you, Patrick.
Thank you, Patrick. Thank you.
So tell us, you've been involved in immunotherapy for a long time now. How did you get started? Maybe we'll start with Dr. Sznol.
I was a fellow at Mount Sinai in 1985, and I think Dr. James Holland sent me up to see a patient who was in, I think, Klingenstein, who was receiving continuous infusion interleukin-2. And it made a mark on me. At the time, I was really more interested in high-dose chemotherapy and growth factors, and I really thought the cure for cancer was going to be bone marrow transplant and giving G-CSF. But as time went on, I became more and more interested in immunotherapy. And actually, when it came time to leave Mount Sinai, there was a job at the NCI to work on immune therapies, and particularly interleukin-2. And because I'd been exposed to it there, I took the job. So that's how I developed my interest.
Great. And in some ways, your interest in bone marrow transplant, which they thought was just a mechanism to increase the chemotherapy dose, turned out to be one of the first immunotherapies. And so that was probably a rightfully placed interest. Dr. Wolchok, how did you get started in immunotherapy?
So my interest started a little bit earlier in my own life, but around the same year as Dr. Sznol's exposure, I was a summer research intern here at Memorial Sloan Kettering with Dr. Alan Houghton and Dr. Lloyd Old. And they were working on one of the first monoclonal antibodies that was being used to treat melanoma by targeting a carbohydrate molecule on the surface of melanoma cells called GD3. And I was charged with developing a pharmacokinetic assay to measure how much of this antibody was in patients' bloodstreams at any given time. For a college student at the time to have, the ability to see truly translational research where, I transported the tubes of blood from the patient's bedside and brought them to the laboratory, studied them, and made rounds with Dr. Houghton on these patients who at the time were having some initial responses to this intervention. It really initiated in me an interest in the field.
Well, let's talk about combination immunotherapies, and especially the combination that you two innovated, the combination of Ipilimumab and Evolumab, which out of thousands of combinations is still one of the only that are FDA approved and has saved so many lives out there. How did you guys come up with this combination?
It wasn't rocket science. There were, I think Ipilimumab, we were working on anti-PD-1 together, actually, at the time. We were both part of that trial. And the only two really active agents in the clinic, immune checkpoint inhibitors, were anti-PD-1 and anti-CTLA-4. Preclinical data were available, particularly from the Medarex group, that combining these two agents could be synergistic. I think Alan Corman presented some of that data. And it was just natural to put this combination together. I was very interested in doing it. I think I approached Medarex at the time that I really wanted to do this trial. And I don't remember the exact details, but I'm so glad that I was paired up with Dr. Wolchok, and we that first part of the trial we did together. We were on phone calls, I think, every week during the initial dose escalation. It was really one of the most exciting times in my my medical career.
Yeah, Jedd, I remember seeing you at a meeting, and you were just so excited. You said, everybody is responding to this combination. So tell us, why do you think the success of this combination, how do you think it's working?
So like Mario, I felt very fortunate to really be in the right place at the right time, on the heels of Ipilimumab having been approved, for the treatment of melanoma. We really started to think about what it could be paired with to enhance the efficacy, and understanding that there was also the potential for increased toxicity. The folks at Medarex and Bristol Myers Squibb at the time, really wanted to have a limited number of sites. And I felt very fortunate to be able to do this along with the team led by Mario at Yale. I think that the idea was that both anti-CTLA-4 and anti-PD-1 converge on the T cell as being the ultimate conveyor of the anti-tumor activity. But it was clear from preclinical studies that the mechanisms by which the two agents got there was non-overlapping. So there was the possibility that there could be additive, if not potentially synergistic activity by combining these two together. We were beginning to learn in the human setting that this might be the case, because some of the first studies of some of the anti-PD-1 drugs, and specifically one of the studies at the time, the drug was known as lambrolizumab, but now we know it as pembrolizumab, were demonstrating that even patients who had, in this case, melanoma that was resistant to the effects of anti-CTLA-4 could respond to PD-1 blockade, again, implying that these were non-redundant pathways. And that really opened up the possibility that there could be further T-cell activation conveyed by adding them together.
Well, having given this combination to many patients and really helping a lot of patients myself, I know there's also some major toxicities with putting these two drugs together. So Mario, you guys have also been the authors of many of the ways to deal with these toxicities. Tell us about those and how you and Jedd figured out how to handle these toxicities.
Well, I think initially, we were seeing the toxicities. Mind you, there was already a lot of information about how to manage Ipilimumab-related toxicities. So by the time we started this trial, all of that prior art had been established by people who had been working with Ipilimumab for many, many years. Jedd is one of them, Steve Hody is another, that really taught us how to manage the side effects of these drugs in the clinic. Rachel Humphrey, who was at BMS, was very involved with that. There were others. Jedd, you might mention the other people who had sort of pioneered the management of these adverse effects. So we already knew how to manage the side effects of Ipilimumab, and the side effects of Ipilimumab and Evolumab were very similar, except just more frequent and sometimes more severe. So on these weekly conference calls, we basically adapted that prior art to managing the toxicity on this trial. I think, if I remember correctly, we pushed a little bit. We really wanted to take these drugs to a different sort of an MTD, because the side effects were, for the most part, reversible. And we were seeing so much activity, we wanted to really push the dose of these drugs to see how much activity we could get from the combination.
That's entirely correct. I would just add Jeff Weber, to that list as well. Jeff was really one of the earlier investigators with anti-CTLA-4 and shared a lot of that knowledge. I remember when I went to one of the initial investigators meeting for Ipilimumab as a monotherapy, Jeff's experience, Steve O'Day's experience, Steve Hody's experience, they were critical to the development of algorithms that we now use to manage these toxicities, which, as Mario said, are they're not unique to the combination. They just happen more often and sometimes more severely in that regard. But I would just add one anecdote, is that the dose escalation for this combination was actually stopped because of a very high frequency of asymptomatic elevations of amylase and lipase. in the dose level that would have been Ipilimumab 3 mg per kilogram and nivolumab 3 mg per kilogram. And so we did treat patients there, and nobody died of pancreatitis, which is what the fear was. But what the experience taught us and taught some of our mentees at the time was to perhaps challenge some of the current dogma around why a dose-limiting toxicity might be a dose-limiting toxicity. And I'll say I take great pride in the work that was led by my colleague, Mike Postow, in challenging the definition of using asymptomatic lipase and amylase elevations as being dose-limiting toxicity when it occurs in the absence of any symptoms of pancreatitis. To the point, actually, that some of the grading systems were actually revised to really distinguish simple lab abnormalities that are not accompanied by symptoms from those that are accompanied by symptoms.
I just want to add, Pat, I just want to add one more person because I hate to miss out people. Ashok Gupta, who was helping us with the trial, we worked very closely with him to rewrite the DLT rules for this trial during the middle of the trial in order to allow us to continue to dose escalate. That's why we had a 50% rate of grade 3/4 immune-related adverse events. But understand that for Ipilimumab 10 milligrams per kilogram, the rate of grade 3/4 immune-related adverse events is in that same range. And almost all of those adverse events are reversible with steroids. So there was a lot of back and forth, I think, during those conference calls that helped shape how we moved forward in the trial. It was really a contribution from all the people that were involved.
Yeah, it's very important. Ashok was extremely supportive.
Yeah. You guys think we're at the right dose right now for this combination? It's been done so many different ways. Higher Ipi, lower Nivo, higher Nivo, lower Ipi. What do you think is the ideal dosing for this combination?
The answer is we don't know.
Yeah.
When we were doing the trial, we saw about the same level of activity in the cohort that used the lower dose of Ipilimumab and the higher dose of nivolumab. But for the quirk of that study, the rate of adverse events in that study, in that cohort, was not lower than the standard dose that was ultimately used in the phase 3 trial. We know later on that flipping the dose, using flip dose Ipi-Nivo, gives us a lower rate of grade 3/4 adverse events. But there's only a phase 2 randomized trial between those two dose schedules. It didn't show any difference in activity, but it was only a randomized phase 2 study. Then when this went on to lung cancer, it was too toxic. And in lung cancer, they started using a much lower dose and schedule of Ipilimumab, 1 milligram per kilogram every 6 weeks, which turned out to be the tolerable and active dose in that setting. So in renal cancer, for example, the flip dose, the lower dose of nivolumab with the higher dose of Ipilimumab, was used in the phase 3 trial. And in a small randomized study, it was just as active as the standard dose. So that's a long way of saying that after all this time, I don't think any of us know what the right dose and schedule of Ipilimumab should be together with nivolumab. Would you agree, Jedd? I
would generally agree. I would add a little bit of additional information, and that's that in melanoma, because as Mario correctly points out, I think the different disease contexts are important in helping to determine what the proper dose and schedule are. So I think that it's important to remember that the study, the randomized trial of flip dose versus the Ipi-3 Nivo-1 regimen, which is what was used in the Checkmate 67, which was the pivotal phase 3 trial, that randomized phase 2 was not designed to be a non-inferiority trial. So we really don't know for certain, if the efficacy is the same. We do know that the toxicity is modestly less when you use the Ipi-1 Nivo-3. If we go back to, basic pharmacology principles, it's important to remember that the dose of Ipilimumab is dynamic between 1 and 10 milligrams per kilogram. The initial studies of Ipi as a monotherapy were quite clear that both the toxicity frequency and the response rate were dependent on dose in that range. And in fact, in a subsequent study that was done comparing Ipi 3 milligrams per kilogram with Ipi 10 milligrams per kilogram, overall survival was better with 10 compared with 3. Yet, for nivolumab, there is no dose dependency in that range. So I think that from my own observations, any dose of nivolumab at or equal to 1 milligram per kilogram at minimum is fine. I don't think that you necessarily gain more by going up on the nivolumab dose. But the Ipilimumab dose is quite dynamic between 1 and 10, and we need to keep that in mind.
Just so everyone knows, the original approval was higher Ipi, lower Nivo, and when they say flip dosing, they mean higher Nivo, lower Ipi.
Right.
So, Mario, you're going to say?
I think there are a lot of scientific questions we don't know. Why is 10 better than 3 for Ipi and melanoma? You saturate probably all the receptors at 3 mg per kg, right? Maybe even a lower dose. So why would 10 be better? So what are the mechanisms that are being invoked by the higher dose that lead to better outcomes in melanoma? Is that because maybe at 10 we're killing more Tregs? Who knows? It's really not known, and knowing the mechanisms would really help us. When you combine with another drug like anti-PD-1, maybe a completely different set of mechanisms are in play, and that may be the reason why you don't need as high a dose of Ipilimumab when you combine with anti-PD-1. But it may be that different patients are different. Different patients respond by different mechanisms. We don't have the tools yet to sort that out in the clinic.
So what do you guys do for your patients? What dose?
Well, go ahead, Jedd. You answer first.
Okay. For a patient that I think is an appropriate candidate to consider the combination therapy, and in general, those are people with obviously stage 4 melanoma, and certainly those who have metastasis in difficult to treat sites, brain, bone, liver. I give Ipi 3 Nivo 1. For people who are perhaps a bit more frail because of comorbidities, or if there's concern of their ability to tolerate the toxicities associated with the Ipi 3 Nivo 1, then I will use either Ipi 1 Nivo 3, single agent PD-1 blockade with any of the approved agents, or now the combination of nivolumab with relatlimab.
We practice identically to that. It's very hard for me to back off from the Ipi 3 Nivo 1 data. That's where the long-term data are. I actually think the activity of Ipi Nivo was underestimated in 067 because patients who had toxicity with Ipi Nivo were not allowed to continue with anti-PD-1 when toxicity recovered. And I think you benefit an additional 5, 6, 10% of patients when you restart anti-PD-1 in that setting. So I have a hard time backing off from the standard dose and schedule of Ipi 3 Nivo 1. I almost give everybody Ipi Nivo in one form or another. I don't treat that many patients with anti-PD-1 alone. And the patients for which, just as Jedd said, for the patients who would be getting anti-PD-1 alone for some reason, I would probably use nivolumab at this point, now that it's approved. So I think we have very similar approaches to managing patients.
Well, you guys developed this combination in melanoma patients, but now it's approved in lung cancer and kidney cancer. And what do you think the future of this combination is in other cancer types?
Well, it works in lung cancer, it works in kidney cancer, works in mesothelioma. I don't even know. Because I mostly focus on melanoma and kidney cancer. I can't even keep track of all the different diseases in which it's been studied. It works in hepatocellular carcinoma, whether standard of care in those diseases or not. I don't know. Jedd, please go ahead. I
Yeah,
can't keep.
no, I think you really covered most of it, Mario. I think that it's also approved for the treatment of some MSI high cancers. I agree with you that it's extremely rewarding to see that, this combination, which, we worked so hard to develop together, in melanoma, is now having favorable activity in other disease settings. And it reminds us, of course, that drug development shouldn't necessarily be histology dependent. There really should be a focus on mechanism. I think that there's, just as there has been in melanoma, there's probably a need to optimize the dose and schedule of this combination across different diseases. It was, of course, very challenging to think about developing a regimen that used different doses and schedules depending upon the disease context. But very happy to see that that took place and that it's happening. I think actually we now are really taking a page out of the textbook that was originally written by the pediatric oncologists in the late 60s and early 70s, when they were first evaluating combination chemotherapy regimens that were providing highly active effects in children with ALL. And once they saw that there were children being cured, but later on, they were noticing that there were toxicities later in life from these very potent combinations. They then focused on refining, the list of agents, the doses of agents, and understanding what's the minimum amount of drug they could get away with that would provide a favorable risk-benefit profile. And I think many of our colleagues are focused on that. I'll just again highlight the work of Mike Postow, who has developed a adaptive dosing clinical trial framework for this combination. And actually, I think it's important for us to remember one of the earliest clinical trials of Ipilimumab was an intra-patient dose escalation study done at the National Cancer Institute, led by Dr. Steve Rosenberg. Patrick, perhaps while you were still there, which went against every rule of cancer drug development, which counseled against intra-patient dose escalation. But I think Dr. Rosenberg, as always, was a real visionary in thinking that there might be a different threshold for both activity and toxicity in individual patients when you're looking at immune modulating medicines. This was, of course, something that was probably first introduced to him during the days of IL-2 development, that different patients could sustain different numbers of infusions of high-dose IL-2. And the goal was to identify in each patient what their risk-benefit profile was. That's a very challenging, but also, in my mind, very elegant way to think about cancer drug development.
Well, after you two were so successful with Ipi-Nivo, the field just exploded with combinations. And in an analysis in February of 2022, it was detailed that over 1,000 combinations have been tried in immuno-oncology right now. However, very few successes with the more recent anti-LAG-3 that you're talking about, Jedd, but otherwise, very few successes. What do you attribute this drought and a low ratio of wins in all of these combinations?
I might be a little bit more optimistic. I mean, you're right that most have not worked, but when you think about combinations that have worked, in addition to Ipilimumab, we've seen combinations with chemotherapy work in a number of different diseases. I would not have predicted that, but that's the case. And now several combinations with chemotherapy are approved. The combinations with VEGFR receptor inhibitors are very interesting. There are approvals in renal cancer and hepatocellular carcinoma. So those were effective, not completely anticipated at the very beginning. And we have, there's some evidence that they can be combined with targeted therapies, but I don't think that's nearly as compelling data as for the other approaches. Where I don't think we have one now proof of concept with LAG-3 in melanoma. We'll see where it goes in other diseases. So I wouldn't say it's been a complete drought. I mean, I think there have been a lot of successes with combinations, although not necessarily IO combinations. So then the big question is, why haven't we seen more successes? We certainly anticipated that all these other checkpoint inhibitors would be active in a subset of patients. Hasn't been the case. It may be that PD-1, PD-L1 pathway is maybe very dominant in humans, but I think the problem is that we just don't understand the immunobiology of many of these tumors very well. And it could be that all these combinations work, but they work in only a small fraction of the patients, and it's hard to detect that in small phase 2 trials.
Dr. Wolchok, what do we have to do to make future studies of IO combinations more successful?
That's a really important question, Dr. Hu. I think that we need to maintain focus on mechanism and be patient. I think that some of the more recent quote unquote failures have been perhaps when there's been a rush toward registration and not enough attention paid to identifying the patient population that is most likely to derive benefit. And again, this is all about understanding the immunobiologic mechanism, as Mario just mentioned. And I think we also have to recognize that as we develop these combinations, of course, as oncologists, we want our patients to improve. That is always our number one goal. But we need to keep our, I would say, our horizons open and broader a little bit, and understand that even when a trial is quote unquote a failure, because there might not be tumor shrinking with a new combination, we need to know whether the agent under development is hitting its proverbial mechanistic target and conveying the immune modulatory signal that it was intended to do. Because what we're seeing more and more of now are trials shutting down early, sometimes after phase 1 or early phase 2 development, because there isn't a robust response rate in patients who have largely been exposed to the standard agents and therefore unlikely to respond to a combination, but without attention being paid to, is the drug even doing what it was mechanistically designed to do? Because there's a fundamental difference between the drug not hitting its target, which is an inferior drug, versus hitting the target, but that not addressing the biology of what's going on in the tumor microenvironment in that individual patient, which is a different challenge entirely.
Well, you two have been such important members of SITC, the Society for Immunotherapy of Cancer, and that focus is on some of this translation and mechanism that you're talking about, Jedd. So can you tell us, you're both officers of SITC, you've contributed so much to the organization. Can you tell us about your roles within SITC and what you think how important that has been for you to be belong to that society, and what the role of the society is in helping to uncover some of this mechanism that we need to understand in order to make future combinations more effective?
Dr. Wolchok, you can start.
Okay. Thanks. I thought we were going in alphabetical order from the beginning, but but I currently serve as the treasurer of SITC, and as such, I'm on the executive committee and the board of directors. I actually have been on the board of directors in the past as well. Quite simply, for me, SITC is the organizational glue that holds our field together, and I think it's an extremely important convener of members of the field from various different backgrounds. So it embraces members who are senior investigators, early stage investigators, those who come from academia, those who come from industry, those who come from the area of drug regulation. And it really serves as a tight-knit community that is laser focused on how we move this field of immunotherapy forward for the benefit of our patients, while, of course, respecting the science that underlies the development of truly effective medicines.
That's a great response. I've been involved with SITC, going way, way back. I can't even remember how many years. It's the only organization where I feel really at home, although I feel at home at AACR and ASCO, but the people who love immunotherapy are in this organization. I was past president, very privileged to work with UPAD and with Jedd and with Lisa Butterfield and many other people in our field. The question is, what can an organization do in order to promote the field? One thing is to provide fellowships and grants. I think one of the things that the organization has done very well is to really promote the career of young investigators, because we need more science, and the only way we're going to get more science is by promoting the careers of people who want to do this work. So I think the organization has played a really critical role in getting people into the field, getting them trained, and getting them out there and working on the problem. We also identify, I think, the major hurdles in the field and trying to address them. How do we do that? By bringing people together, as Jedd mentioned, holding meetings and nudging the field along in certain directions. And I think we've done that very effectively. It's just, I think, been incredibly successful, and we still do that. I mean, we hold meetings that are critical for the field. We've had meetings on cytokines and on surface targets and on T cells. We hold deep dives that help educate people at a deep level about individual targets. We bring the FDA, industry, academic people together to address major problems, sometimes regulatory problems that are a hurdle to the field. I just think this organization does incredibly important work, and I'm privileged to have been a part of it.
You guys have been such great mentors also to the next generation, because you're right, it's all about getting that next generation ready. What advice do you have for that next generation?
I think it's really about being patient. We live in such a fast-paced world. I know that sounds like an overstatement, but one thing that I hear from some of the folks that I'm very privileged to mentor is that they entered the field because of the success that they rightfully saw during the early days of PD-1 and CTLA-4 and then combinations, and they thought that they were jumping on the express train, to career success, and that was a very logical assumption. But I think what I would counsel them is not to be disappointed if that train has to slow down occasionally, because I think those of us who have a little bit more gray hair and remember when SITC was called, SBT or ISBT, C, or, those were years when cancer immunology was really not a field that many people wanted to hear about. It was a field that was by some folks considered to be speculative science. We went through some very difficult times when, frankly, people didn't believe what we were trying to suggest. And so if you take a snapshot of the field as, starting in 2011 with the approval of Ipilimumab and continuing to now, you would think that this is, really nothing but a series of successes. But the truth is that science is a pattern of ups and downs. And that's the nature of the beast. And we ride through that and we learn from our failures.
Dr. Sznol, what's your advice for young investigators?
Dr. Wolchok's advice is great. The patients, continue to, pursue your dreams, your ideas. I think what's different today than maybe when we started on the field is the science is so amazing. right? There's so many things that people can do. Every day I see these feats of, protein engineering that are amazing. You can just create these amazing compounds. So I agree. Not everything is going to work. We have to be a patient. We have to keep trying. But gosh, I wish I could start all over again, because I just think the tools are amazing. The clinical trials are amazing. I see, 10 different drugs every day I'd love to study in the clinic. It's an incredibly exciting time. Um, I would say stick with it. It's going to be a wild ride for the next, I think 10 or 15, 20 years.
I agree. It's going to be so fun for these young investigators. And you're right. When we started out, we knew nothing compared to what they know now, with all the mechanistic actions of all these checkpoint inhibitors, T cell therapy, CRISPR. I mean, it's really going to be incredible. Now, Jedd, let's talk about your most important role, and that is as a tuba player for the checkpoints. That's incredibly important. It's what we call the heavy metal section of the band. Did you ever think in your tuba playing days that you would be in a rock band?
Not really, Patrick. And I have to say that it is such a joy, to be part of that group.
We're going to be at the annual meeting in November, a SITC annual meeting, which is going to be in Boston. So, Mario, you coming to the Boston meeting?
I absolutely will be at the Boston meeting, and I'm very envious that have no musical talent and can't be in the band. I just feel, I need to go back and take music lessons so I can be in the Checkpoints band. It'll take me about 10 years, but
well, we could add a kazoo player maybe. Jedd, you want to add a kazoo player?
Yeah, absolutely. Always room for a kazoo.
Yeah, that's right. So, well, Boston is a wonderful town. I hope to see many people at the meeting this November in Boston. Tell us about your cities, though. New York City versus New Haven. Now, both have their advantages and their disadvantages. Which one's better to live in? Jedd, you go first.
I'm a very subjective, responder to this question, having, spent most of my life in New York City. I mean, this is obviously it's a world-class city, very diverse, very culturally rich, very scientifically rich, with outstanding, medical institutions. But it's sometimes it's a hard place to live, right? It's sometimes if you want to think about, like driving somewhere, you don't just jump in your car and go, right? There's a lot of planning that has to take place. Thankfully, we have wonderful public transportation. I think that, like many cities in America, we have, the serious problem of income disparity to deal with, that we face every day. During my training, I had the real privilege of spending about 5 years training at one of the world's most eminent charity hospitals, Bellevue Hospital, part of the New York City Health and Hospitals Corporation, and got to serve the population of New York regardless of their ability to pay. And I think that's one of the things that makes New York wonderful in my mind is the things that are provided to people who don't have means that would not necessarily be provided elsewhere. And so I think every time people gripe about the taxes that New York may charge, I usually counter with, yes, but we have a safety net hospital system that provides academically supported care to the poor. And it's great partnerships between our academic hospitals, in the case of Bellevue, New York University Medical Center, and Bellevue, but certainly many of the other academic centers in New York that have partnered with the safety net hospitals.
All right, Mario, why would anyone want to live in New Haven?
Pizza.
There's a little bit more here than pizza. I love New York City, by the way, and New York City is only an hour away by train. So the New Haven's really a charming small town. It's the home of Yale University. you know, you feel like you're back in college again. When you walk around downtown, you feel energized by all the students, all the people that are here. Still a very sophisticated city with amazing museums, and I wouldn't say our restaurant scene is as good as New York City, but it is a small town. There's a lot of great things here. And like Jedd, living in Connecticut is great because I have to say, in all the years that I've been here, I've never had to refuse a patient therapy. We've always been able to find, you know, treatment for everybody, regardless of their means. And it's really nice to work in a place where you know that if somebody walks in to your clinic as referred to you, you're going to be able to treat them with state-of-the-art therapies, regardless of where they came from or how much money they have. So it's nice to work in that environment. They're great people, great colleagues. It actually is a great place to live. Not quite as busy as New York, but still a great place to live.
Well, I really want to thank you guys for your time today. Anything else you'd like to bring up?
Just want to thank you, Patrick, for first of all, for your leadership, for your keyboard skills, right? And for bringing us together in this fireside chat. I just wanted to know, like, are there going to be backgrounds with like roaring fires, you know, behind us when this eventually goes to air?
We can put anything in that you want to. We have all the technical abilities possible. Well, thank you to Drs. Mario Sznol and Dr. Jedd Wolchok. These are two superstars in the field of immunotherapy, but they're also two of the most humble people that I know. So they've contributed so much and saved so many lives, both in their personal clinics, but also in the therapies that they've developed that have gone on to treat many melanoma patients, but many other kinds of cancer as well. So thank you for your contribution, Drs. Sznol and Wolchok, and thank you for your time today.
Thanks, Pat. Thanks for bringing us together. It's a great pleasure to work with you. Thank you.
Always.